All right, guys, we are back in business. Okay, so we talked about cloning in general in class, which dealt with whether or not we should actually use our cloning for Dolly sheep to spread it to mammalian human cloning. And this is going to raise a lot of ethical conditions and issues. Really, in order to do cloning, we should take a look at it in terms of its therapeutic method as opposed to just taking an entire human species, us, and cloning us all together. We should actually start to use it as a form of therapy. Therapeutic cloning is basically an ability to take embryonic stem cells, and these cells are used for a number of therapeutic purposes. For example, using cancer research, if we pull it from the embryo itself, and then in this case, it's going to ensure that it'll give rise to several cells later on in life. And so it will typically only give rise to a number of limited range of cell types. But again, it's much easier to give rise to these differentiated cell types and use for a variety of therapeutic purposes. For example, bone marrow transplants, folks who have extreme types of cancer, that then they can use therapeutic cloning research to actually ensure that they're not going to get sick from that cancer. So one way we can use therapeutic cloning instead of adults, human embryonic, what's it called, just cloning in general, is to do this. If we take that blastula and we take the embryonic stem cells from that and we remove the cells inside of it, what you're going to do is culture those cells, those embryonic stem cells, and then we take, for example, the adult stem cells and bone marrow, and we culture all of these in different conditions, right? So once we do that, we can get blood cells, we can get nerve cells, and we can get different types of differentiated cells. And this is really a huge benefit for cancer research because you can use this to transplant it back into a patient with leukemia. I know Texas MD Anderson had a huge case where there was a young 55-year-old, very young comparatively speaking, in the prime of his legal career, had basically gotten bone marrow cancer, which is essentially leukemia. So as a result, they did some stem cell research on him and injected it back into his bone marrow, and now he actually is 100% leukemia free. So this is one example of therapeutic cloning, and this is a couple of years ago where this happened. So we're talking about something that has got above and beyond a huge amount of therapy, especially for those folks with types of leukemia or bone marrow cancer. Okay, so in nature, how do embryonic stem cells differ from adult stem cells? So embryonic refers to those that are still sort of pulled from the embryo itself. So it gives rise to different types of cells in the body, whereas adult stem cells only give rise to certain types of cells that are not, particularly every type of cell. So that's why we like to use embryonic stem cells, for example, in embryonic cord blood, and not necessarily just adult stem cells, because those don't really differentiate into lots of species. These guys just differentiate into embryonic stem cells, into anything you want. Like we said in the picture before, into muscle, into nerve, or even in this case, bone marrow, which is going to sort of therapeutically help that patient. So this leads us directly into what cancer is. A very important part, I would say, besides cloning and cancer. These are the two most huge topics today in cancer research. So we have to look at how exactly cancers can start. So cancer cells, which, like I said, we talked about this earlier, actually divide mitotically in a very uncontrolled state. So they go awry. They usually result from mutations in genes. And so that mutation can change into what we call a proto-oncogene, which is a normal gene that helps control cell division and is typically going to change into an oncogene. So proto means before, right? It was something that was normal that helped control cell division, and continues to divide, essentially turns into an oncogene, which unfortunately starts to divide excessively. And this is what usually causes that uncontrollable type of mutation. Any leukemia, lymphoma typically has an oncogenic response because it's going to divide excessively. And so you normally hear about cancer that is in remission. If it is, it's therapeutically held down by the T cells and the B cells inside the body. So you're relying on those helper immune cells to target the actual spread of the cancer. And then, of course, you can do chemoradiation, excise the tissue, and do immunotherapy, right? But if that oncogene starts to divide excessively, your cancer may actually come back. And so this is the really scary part about cancer. We just don't know when it's in remission. If the health of the patient is actually poor, cancer can actually come back. So mutations that activate tumor suppressor genes may also lead to cancer. Tumor suppressor genes are again inside the body. These are types of genes that can also lead to cancer. And so certain mutations can inactivate them, and as a result, this can lead to very dangerous levels of cancer. So here's a earlier version of an oncogene that stimulates normal cell division. What can happen is a couple of things where you can have a mutation within the gene to activate the oncogene, and this hyperactivity can stimulate a protein in a normal amount. Another thing is you can have multiple copies of the gene, so many versions of it. This normal growth will stimulate the protein in excess, or you can have a mutation within a control region of DNA where the promoter is going to start, and then you're going to have normal growth stimulating the protein in excess. So there's many ways to make oncogenes, which all lead to really excessive cancerous mitotic growth, all very bad ways to have uncontrollable cell division or cancer. So what happens in a tumor suppressor gene is the normal growth inhibiting protein will have cell division under control. So if you have this tumor suppressor gene mutated, it'll have a defective protein, and unfortunately it will create this kind of mutation where you're normally going to see a couple of cells undergrowing absolutely rapidly, and this is exactly where it's not under any control. So this is when that cancer continues to spread. So like I said, cancer can undergo remission, or in this case, it can be exposed to that essentially the mutated tumor suppressor gene, and as a result, it can grow uncontrollably. So multiple genetic changes can underlie the development of cancer. Cancer can result from a series of genetic changes, or colon cancer itself can illustrate the gradual progression from a type of mutation to cancer. So, for example, if you have early exposures to carcinogens, you smoke a lot, you drink a lot, this is going to be a greater risk for, if you do it earlier in life, it's going to be the same exposure. This is going to essentially carry greater risk than the same exposure later in life. So if you do it early on, it's got more time to accumulate additional changes to it and potentially lead to disease. So, for example, my boyfriend, he dips. And so I always tell him this is going to lead to earlier, mouth cancer, and it's going to cause mouth cancer later on in life. And it's just one of those things I guess just doesn't care about, right? So, for example, in your DNA, your DNA itself, you have an oncogene which is activated, right? And unfortunately, cellularly, this can increase division mitotically throughout the body. If this tumor suppressor gene gets inactivated, that stops the suppression of the tumor. If it's inactivated, your polyp can grow, and that polyp can invade the colon wall and unfortunately create colon cancer, right? So this is typically how colon cancer is created. So that's why when we do a colonoscopy, it's very important because the physician goes in there and he just excises these small polyps. Now, polyp growth can occur in a 20-year-old individual. It grows on early on in life. It's just one of those natural things that's inside the body, and so it typically is you have to excise it, remove it, because if you do not have the colonoscopy screening, every couple years, You have to follow what your doctor's guidelines are, 3 to 4 years if it's been perfectly healthy, or if it's, you're much older, you should do a colonoscopy every 2 years, right? If you've had colonoscopy or colon cancer before, you should probably do the scanning every year. So that's something that your physician will determine is the correct schedule to prevent things like colon cancer, right? It's one of those easy cancers that you can prevent because, you do the colonoscopy, you scope the patient, he takes that or she takes that medicine, which ensures that they are basically cleaning the colon. You get rid of all the junk. It's a clean, healthy bowel. And then you're able to scope it and observe every area, every wall of that colon in the large intestine. Once it gets to this ileocecal valve, you can stop and basically say, all right, we got a healthy colon. As soon as they start to see a polyp, they're going to excise it. So it's like they're cleaning it as they go along too, okay? So don't just assume that they're just putting the tube up there and scoping you and, nothing's really going to be done. They are excising the polyps as they go along. So here's a couple of accumulation of mutations. So if you have four different types of chromosomes, the first mutation, the second mutation, the third mutation, the fourth mutation, these constant mutations will lead to a malignant, very dangerous cancer cell, and this is exactly how cancer starts to spread. So cells themselves, like epithelial cells that cover and line organs and glands, including your stratified squamous epithelium, which is keratinized, full of basically layering the largest organ in the body, your skin. Those that line body cavities they're frequently replaced, and so they divide more often than most other types of body cells. So the question is, will epithelial cells become cancerous more or less frequently than other types of cells? And if you think about it, because they are constantly replacing each other, you have increased chances that you're constantly getting more frequent mutations. So you actually have a greater chance of cancer in the epithelial region than anything else. So you hear a lot of melanoma, epithelial cancers in the body. Okay. So there's a lot of faulty proteins too that can interfere with normal transduction pathways, and we talked about this earlier today. So for example, signal transduction can lead to the synthesis of proteins that influence the cell cycle. So many of these earlier proto-oncogenes and tumor suppressor genes, they can code for proteins and that are active in signal transduction pathways and that regulate gene and cell division. So for example, there's a RAS level of genes that typically start to have, in this case, increased chances for cancer or mutations to exist. So if you have a normal RAS gene, right, this normal RAS gene is going to give you normal cell division. If you don't have a growth factor, we talked about growth factors which manipulate genes themselves. If you don't have a normal growth factor, this is not going to produce a normal RAS gene. And unfortunately, you're going to have a lot of overexpression of the protein, which can increase cell division and actually change how an oncogene is formulated. So this is another way that you can actually develop cancer. So there's a normal growth inhibiting factor that's undergoing this cell-to-cell signaling cycle. It transduces. You got a normal p53 product, and so this is going to activate and undergo normal cell division, transcription, translation, right? There's no cell division as a result, and so this is an inhibitory cell signal transduction pathway, and this is usually going to affect a faulty tumor suppressor protein. So here's the mutant. This is the normal one. The mutant will then undergo a non-functioning transcription factor. It will not trigger that. It will basically not trigger transcription, and as a result, no translation will occur. The protein will be absent, so this is going to increase cell division. So this is a faulty tumor suppressor protein, and this guy here, you have no cell division. You can stop, in this case, the cancer from going. So contrast the action of an oncogene with that of a cancer-causing mutation in a tumor suppressor gene. So what's the difference between an oncogene? Well, an oncogene normally encodes an abnormal protein, and this will stimulate cell division through a signal transduction pathway. A mutant tumor suppressor gene, this guy will encode a defective protein that can't function in a pathway, and this will stop cell division. So those are the two differences between it. And, here in the United States, most industrialized countries, first world countries, these guys typically will have cancer, and then the other one is heart disease and heart attack and stroke. But cancer is the second leading cause of death after heart disease in most industrialized nations. Most cancers arise from mutations caused by environmental factors, for example, carcinogens. These are agents that alter DNA and make cells cancerous. Unfortunately, you could change your lifestyle. You could stop smoking, you stop drinking, stop dipping. This can basically reduce the chances for cancer to initiate inside the body, right? So this is another thing like skin cancer. A lot of people who are young, I remember when I was in high school, a lot of girls used to go to the tanning bed to try to get darker. This obviously, because they're younger, will expose the epithelial cells to the light and the sun, and the UV and UVA and UVB will cause cancer later on in life. So you're not going to get it early on. You're probably going to get it later on in life. So here's a checkpoint here of all of the data presented. This is 2015, the deaths in thousands. So here we have testicular and cervical, oral, liver, and ovarian cancer, pancreatic cancer, a huge percentage of that. We now have thyroid, uterine, leukemia, lymphoma. So we're getting to the scarier types, right? There's the skin and the blood cells themselves are lymphoma. Colorectal, again, you can start to prevent that through colonoscopy and try to catch those polyps from actually mutating and adhering to the wall of the colon itself or the GI tract. Breast cancer, prostate cancer, lung cancer. This guy is the highest causing. Here's the risk factors: alcohol, diet, female, women typically have uterine and cervical cancer, genetics, if you're male, obesity, pollution, radiation, smoking, sun exposure, STIs or STDs, that's sexually transmitted infections or sexually transmitted diseases, and then viruses. So this is going to increase the fatality rate if you have all of these risk factors. So that's one of the first things they ask you. You're at the doctor's office, they'll ask you, do you drink, do you smoke, and then they ask you frequency, right? And they'll also ask you, how many times have you had unprotected sex, or how many partners have you had in the last, a year, which is unprotected. That's another increased risk of STIs or STDs. So especially in this population of college kids, you guys should be more familiar and educate yourself on types of STIs and STDs. We're not just talking about gonorrhea, syphilis, but we're talking about cervical cancer and ensuring you have the intelligence behind you before you do get involved in that you know what exactly your partner has, and you have a conversation with them. You use protection. Ladies, don't rely on the guys to bring the condom. Focus on yourself. Walk out that door if he does not bring the condom. So that's straightforward. Take control of your reproductive health. And I argue this because I just hear about more and more young kids who are just engraved with tons of STIs and STDs that we just didn't have 5 to 10 to 15, 20 years ago, right? So today's population, we're taking a look at cervical cancer, uterine cancer, HSV1, HSV2, that's herpes simplex virus 1, which is oral or mouth cancer, and HSV2, which is herpes simplex virus 2, which is anal genital cancer, right? Or excuse me, anal genital herpes. Be smart and protect yourself, before you engage in sexual activity. Okay. All right. So this is the end of the chapter. You guys should basically be able to kind of compare all of these. We don't need to do this. We actually took this out, the LAC operon, the TRP operon, and different operons that are used to activate. But we did talk about a selected gene expression, how DNA is kind of packed into those 8 histone proteins, which are wrapped in a nucleosome, which is then highly coiled in the chromatin, which condenses in your Barr bodies in the chromosomes, right? And then explain how that cat's tortoiseshell appearance formed, and why that sort of orange and black fur appeared, especially for that tortoise shell. So tell me about X-linked inheritance as a result of that. And then explain this to your fellow lab partners. And then this should be one way to help understand if you got chapter 11 nicely. All right, good job, guys. So we finished off. We're going to go ahead and do chapter 12 on Monday. So I'll see you guys then, and have a good rest of your Monday.